Hello everyone. We are fourth year biomedical science students. Here in the Biomed Club channel, we are going to give you a stream of videos regarding various bioinformatics concepts. To begin with, in this video, we are going to show the general overview of NCBI and how it works. Getting into NCBI, first step is to search www.ncbi.nlm.nih.gov or NCBI in Chrome and click on the first link which appears. After that, there will be a home page of NCBI. Now, NCBI stands for National Centre for Biotechnological Information. The NCBI houses a series of databases relevant to biotechnology and biomedicine, and it is an important resource for bioinformatic tools and services. As a national resource for molecular biology information, NCBI's mission is to develop a new information technologies to aid in the understanding of fundamental molecular and genetic processes that control health and disease. It is a comprehensive website for biologists, including biology-related database tools for viewing and analyzing the medical data automated system to analyze information where strong and retrieval of the medical data is also possible. It covers all the biotechnological information, biochemical, biotechnology, chemical information about living organism and all the species. Hence, all the data discovered about the new species is also stored on NCBI. Now, once you are in the home page on the NCBI, left side where there will be a list of resources that you have to access, and at the right side there is a list of most popular resources used such as BLAST tool or PubMed. And in the uppermost middle part of there is a search bar and a scroll down for all databases option. To get detailed information, let's take an example of hexokinase enzyme in the search bar. For now, let it all be database selected and click on the search bar option. Here it shows results shown in the different databases as we have not particularly selected any one database. There are different databases are literature, genes, proteins, genomes, clinical, and PubMed. We are provided after search. Literature. It has different portals like Bookshelf, Medical Subject Heading, NLM Catalog, PubMed, and PubMed Center. These portals are used for finding the information of protein, gene, nucleotide, etc. as we search for. It provides information in the form of books, medical literature, abstracts, articles, etc. Second is genes. It has different portals like Gene, GOE database, GOE profiles, homologene, and PopSet. These portals provide various information related to genes such as its location, phenotype, expression, etc. Third is proteins. The different portals over here are conserved domain, identified protein groups, protein family models, and structure, which would provide details on its locus, origin, its FASTA sequence, other identical proteins, and many more. Next is genome. Here are the different portals are assembly, biocollection, bioproject, biosample, genome, nucleotide, SRA, which is sequence read archive, and taxonomy. It shows information on different nucleotide sequences, projects, and other related samples. Next is clinical. The different portals over here are clinicaltrial.gov, CleanWire, Database GAP, Database SNP, GTR, MedGen, and OMIM. It shows information regarding the relationship between genotypes and phenotypes. Also, here user can search, view, and download data from submitted studies. Last is PubChem. Here, the different portals are bioassays, compounds, pathways, and substances which show concerns about the potency of biological products.
So now going back, as we can see three different sections. On the left side, there are many different types of resources present. It starts with the list of resources on the top. It is an alphabetical order of different databases which can be accessible to us. If we click on all resources, then things like assembly, biocollection, and bioproject are present. Then if we go further down on same page, there are GenBank databases of phenotype and genotype, and many more databases are available. There are chemical and bioassays where we can learn about different chemical use. There are DNA, RNA, data and software, and many more uh, resources where we can explore various databases. Then if we click on sequence analysis, there we can learn about how to run BLAST or FASTA, which is used to compare sequence of different organisms. Then there is also a literature option available where you can find different research paper or books related to your topic of interest. Moving further down, we can see training and tutorials where we can learn about how to work on NCBI. This all resources are free and accessible. You can explore more, which would help you to get a better understanding on how to work on NCBI. Now, coming on the center of the page, we can see 6 different icons. The first is the submit. If we click on a submit, a portal will be open. In this portal, we can type a few words about the sequence data we are submitting and select an option to learn more. Different tools are included for the submission in the portal like GenBank, Sequence Reading Archive, which is SRA, GEO, TEO, etc. The benefit of submitting the data is that it will make your sequence data available in the International Nucleotide Sequence Database collaboration. Then going back on the front page, the second one is the download. So the majority of data on NCBI are available for the download either directly from NCBI or FTP site or by using software tools to download custom data sets. Software tools are ESPER, which are used for high-speed download of data. Moreover, NCBI provides several tools for downloading custom data sets, which are entries, program utilization, and SRA toolkit. The third one is learn. So here NCBI creates a variety of educational products including course, workshop, webinars, training materials and documentation. NCBI is educational events are open and free to everyone. All the NCBI education materials are available for anyone to reuse and distribute. For example, if we click on webinars and course, there will be a list of different types of webinar which are going to take place in the future and which you can enrollment into. This all different events or webinars are conducted by NCBI for better learning of it. Then going back, the fourth one is development. Here, NCBI provides various resources that allows us to access and manipulate NCBI data in their application. NCBI uses resources such as APIs, code libraries, and data formats are used for the development. Then the next one is analysis. Analysis provides a variety of data analysis tool that allows the user to manipulate, align, and visualize and evaluate the biological data. Then going back again, the last one is the research. In research, the NCBI Computational Biological Branch, which is CBB, focus on theoretical, analytical, and applicative computational approach to the broad range of fundamental problems in molecular biology and medicines.
Now, on the right hand side of this NCBI homepage, various popular resources are given. From it, let's see the first, that is PubMed. So basically, PubMed is a free resource supporting the search and retrieval of biomedical and life sciences literature with the aim of improving health both globally and personally. The PubMed database contains more than 34 million citations and abstracts from biomedical literatures. However, it does not include full text journal articles, but links to the full text and are often present when available from other sources such as the publisher website of PubChem Central, that is PMC. Now we'll see the second resource, that is bookshelf. So again, bookshelf is an online resource providing free access to the full text of books and documents in life sciences, healthcare, and built and maintained by the NCBI with the collaboration with National Library of Medicine, that is NLM. So basically bookshelf complements the other biomedical literature resources also, like PubMed Central, NLM Catalog, and various other, which gives other materials and also audio visual sites. Then the third is PMC, that is PubMed Central. So PubMed Central is a full text archive that includes articles from journals reviewed and selected by NLM for archiving, that is current and historical informations, as well as individual articles collected from archiving in compliance with funder policies. Now, the fourth is BLAST, that is BLAST, the basic local alignment search tool, which find regions of local similarities between sequences. This program compares a nucleotide or protein sequences to sequence databases and calculates the statistical significance of matches. This database BLAST can be used to infer functions and evolutionary relationships between sequences, as well as can help in identify the number of gene families. This tool can be described in detail in our coming video links. Now, the fifth database is the nucleotide database, which is the collection of sequences from several sources, including gene bank, reference sequence, TPA, and protein database, that is PDB. This gene and transcript sequences data provides the foundation for biomedical research and discoveries. Then the next is NCBI's genome resource. This includes information on large scale genomics projects, genomic sequences and assemblies, and mapped annotations such as variations, markers, and data from epigenomics studies. It also includes collections of curated nucleotide sequences used as references, sequence clusters to predict and study homology between the data, and various databases and tools for the study of gene expression. Then is the single nucleotide polymorphism, that is SNPs, which is pronounced as SNPs. So these are the most common type of genetic variations among people. This database SNP contains human single nucleotide variations, microsatellites, and small scale insertions and deletions, along with population frequency, molecular consequences, and genomic and reference sequence mapping information for both common variations and clinical mutations. Then the next one is NCBI's gene resource, which includes collections of curated nucleotide sequence used as reference sequence cluster to predict and study homology, and various databases and tools for the study of gene expression. These database would also be informed in detail, described in detail in our coming video links. Then the next is the protein database, which is the collection of sequences from several sources, including translations from annotated coding regions in gene bank, reference sequence, and TPA, as well as records from SwissPort, like other protein database, like other databases. Then these protein sequences are the fundamental determinants of biological structure and functions, hence provides an primary and valuable information on proteins. Then the next is PubChem. So PubChem is a public repository for information on chemical substances and their biological activities, which was launched back in 2004 as a component of the Molecular Libraries Roadmap initiatives of the US National Institutes of Health, NIH. So this PubChem is the largest open source chemistry database, and hence is an important database when it comes in terms of drug discovery, since it contains active chemical substances from a variety of sources, which increases the probability of successful lead identification. So now coming back to the NCBI homepage, on the right hand side below popular resources is the column of NCBI news and blog. This NCBI Insights blog is intended to help you to better understand the NCBI and the resources, explore issues of scientific interest that drive your resource development, and demonstrate how can you use their resources to help enhance your research. Moreover, any updates or any new versions available in this portal is notified here. Below it, on the right hand side corner, click on the more. It will open the portal of NCBI Insights, which is a complement to their existing education and outreach programs, social media outlets, and help desk user support. If you have any doubt on topics or suggestions for blog, so you can post it here. So now coming back to the NCBI homepage, below that is COVID-19 information, which gives information about COVID-19 pandemic, SARS COVID-2 data by NCBI, and any other prevention and treatment information by HSS, and many more. Below that is about the links from which we can follow NCBI, like Twitter, NCBI Digest Twitter, Facebook, LinkedIn, and various other social media links. Hope myself, Bhargavi Patel, and my partners Kaksha Shah and Disha Sheth has provided you with sufficient and helpful information on general overview of NCBI. Thank you.